This presentation is about biotin. Biotin is a water-soluble B vitamin. The discovery of biotin occurred while researchers were trying to find a cause for egg white injury. Egg white injury was caused by the excess consumption of raw eggs. A protein in egg whites, known as avidin, binds to biotin and inhibits its absorption. In 1931, Svent Georgi discovered a substance that prevented and treated egg white injury. In the 1940s, Kogel and Duvigneau identified that structure as biotin. Biotin structure contains two combined rings, a uridyl ring and a thiophene ring, with a valeric acid side chain. Biotin functions in the body as a coenzyme, which helps transfer carbon dioxide to different macromolecules, and also functions as a non-coenzyme. HLCS, also known as halocarboxylase synthetase ligase, is the gene responsible for producing the enzyme halocarboxylase synthetase. This enzyme helps bind biotin to other carboxylase enzymes in order to activate their function. This plays a major role in how biotin is utilized in the body. The covalent bond between biotin and an enzyme, protein, peptide, nucleic acid, or other molecules is known as biotinylation. Coenzyme roles of biotin. There are 4 biotin-dependent carboxylase enzymes in the human body. These enzymes are key players in metabolic pathways such as gluconeogenesis, amino acid breakdown, and fatty acid synthesis. And these enzymes are pyruvate carboxylase, acetyl-CoA carboxylase, propionyl-CoA carboxylase, beta-methylcrotenyl-CoA carboxylase. Biotin also facilitates glucose storage by stimulating the enzyme glucokinase. Pyruvate carboxylase is an essential enzyme for gluconeogenesis. This biotin-dependent enzyme helps convert pyruvate to oxaloacetate for the purpose of replenishing the TCA cycle. Acetyl-CoA carboxylase is involved in the synthesis of malonyl-CoA by combining acetyl-CoA with bicarbonate. Malonyl-CoA is an important enzyme for the synthesis of fatty acids. Propionyl-CoA carboxylase is another important enzyme that breaks down the amino acids isoleucine, threonine, and methionine into propionyl-CoA. Propionyl-CoA is also carboxylated to dimethylmalonyl-CoA, which converts to succinyl-CoA, an important part of the citric acid cycle, and is also involved in the synthesis of heme. Beta-methylcrotenyl-CoA carboxylase breaks down the amino acid leucine to beta-methylcrotenyl-CoA. Beta-methylcrotenyl-CoA then reacts with ATP and magnesium to produce acetoacetate and acetyl-CoA. Regulation in the body. In food sources, biotin is typically bound to proteins. SMVT, known as sodium-dependent multivitamin transporter, carries biotin across the brush border during absorption. During digestion, the enzymes pepsin and proteases within the intestine break apart biotin from the attached protein. This is necessary for biotin to be absorbed. The breakdown of this compound produces free biotin, biotidynyl peptides, or biocytin. The biotidynyl peptides continue to be broken down by enzymes in the small intestine. The biocytin also needs to be broken down continuously into free biotin and lysine. Food sources of biotin include eggs, avocados, salmon, liver, raspberries. Adequate intake is recommended for biotin. Biotin deficiency is rare because the bacteria in the colon is able to synthesize a small amount, and also biotin is rarely available in foods. Deficiency can occur with excess consumption of raw eggs in those with GI diseases or long-term use of antibiotics. Genetic diseases that affect the gene HLCS or the enzyme biotinidase. Some signs and symptoms of deficiency include neurological, like depression, lethargy, hallucinations, dermatitis, hair loss, grayish pale skin, impaired immune system, anorexia, increased serum cholesterol. Toxicity has not been seen from consumption of biotin greater than 100 milligrams per day. Plasma biotin can be assessed with normal levels within 215 to 750 pg per ml. This type of testing may not be a reliable test to check for overall status of biotin in the body. Checking urinary biotin may help detect early onset of biotin deficiency. This test detects organic acids, in particular 3-HIA excreted in the urine. Normal levels of 3-HIA are less than 0.2 micromoles per milligram creatinine. Deficiency is less than 6 micrograms per D or 200 pg per ml. Drug interactions. Anticonvulsants can interfere with the absorption of biotin in the small intestine, which then will contribute to low levels of biotin in the plasma. Antibiotics or sulfa drugs may stop biotin synthesis in the colon due to killing off the intestinal bacteria. Clinical correlations. During pregnancy, biotin is increasingly broken down, and therefore 300 micrograms per day of biotin in prenatal vitamins may be helpful. Biotin may also help treat dermatitis, brittle nails, and diabetes mellitus.